basic concept, make sure that you kind of take a look at it and that you're comfortable with the basic premise. But what we'll talk about here in just a second is a couple different ways that this works. All right. Now, one of the things that's interesting is, in case you were wondering, right, where did these antibodies come from? These known antibodies. Basically, what you can do is, in some cases, you can inject an antigen into a rabbit, right? Rabbit's one of the common animals used, so are mice. You inject your antigen of choice into them. They generate an antibody response, all right? You then isolate B cells from them, and you identify B cells that have been generated in response to the antigen you previously injected. And you basically merge that B cell with a cancer cell. It's called a hybridoma. And then you have a B cell that essentially produces an unlimited amount of antibodies, all right, that you can then isolate and use in research. All right. The problem, again, with this, or not necessarily a problem, but because it's polyclonal, basically what that means is, if you remember way back when, right, probably a couple of weeks ago, when we had that picture of antibodies binding to different antigens on the same bacteria, that's what you're kind of getting, you're getting here, right? And so it's going to bind, you can have multiple antibodies that could bind to the same thing. And in some cases, these antibodies might bind to things that are related, but not the actual thing you're looking for. All right. So from a research setting, these are very powerful because they cover a wide range, but they're kind of a little sloppy or dirty. All right. And so here's actually, I'm sorry, I got a little ahead of myself. Here's what I'm talking about when I talk about the using cancer cells to make unlimited antibodies. So here, right, you immunize a mouse. Okay. An example of an unknown antibody. Yeah, so like an unknown antibody, what that would be is if you had a pathogen that you were interested in. So I'll use the example. So what I used to do in my research in grad school is there is a protein called CBIR, CBIR, and it's actually a protein that's part of the flagella in bacteria that can be in the gut. And what I would do is I would take serum from a mouse. So I'd take blood from a mouse, and then I would look for the presence of an antibody to that CBIR protein. And the way I would do that, as you'll see here in just a second, is I would coat a plate, again, you'll see what this looks like in a second, with the CBIR antigen. And then I would take the blood sample from the mouse, which has a bunch of antibodies in it, and then I would look and see if I could find an antibody that would bind to that antigen. So when I say an unknown antibody, right, you still have to have, again, that target that you're going after. So I hope, does that make sense, Lauren? Okay, good. Ooh, sorry. So in this case, this is what I was talking about for the, I'm sorry, I got these mixed up. So if you go back here, right, this you're just injecting, again, the whole bacteria. So that's why you have this mixture into the organism. And then you're just pulling antibodies from the serum of this mouse or this rabbit. All right. Here, this is where you get more specific. So this is monoclonal antibodies. So in this case, you inject the mouse with an antigen. All right. And so just like before, right, even with the rabbit, you have B cells that respond to different parts of that pathogen that you've injected. All right. what you then do, right, is you actually fuse these B cells with these melanoma cells, right, these cancer cells. Remember, cancer cells basically have unlimited growth potential as long as you give them nutrients. So when you merge these, that's when you get this hybridoma. And so you now have basically a modified B cancer cell that can produce all these antibodies. And what you can do, though, is it's easy enough in a research setting, is you can actually isolate these individual cells and you can get them to produce, right, their specific antibodies. So then you have, instead of having a mixture of antibodies, you have individual antibodies that you can then use to study your organism. These are a lot cleaner because it's very specific to an antigen from a pathogen, all right? These are harder and more time consuming to generate, but typically if you had a choice, you'd want to go with a monoclonal antibody in a research setting, all right? So, you know, how do we then identify the pathogen antibody interaction, right? And what we can do with these antibodies that we generate, right, is that we can make antibodies to the antibodies that are labeled. And again, you'll see what that looks like in just a second. We can label these with enzymes that will cleave chemicals to change colors of solutions. We actually have fluorescent dyes. So if you want to see some cool pictures, look up fluorescent histochemistry. And we have radioactive isotopes that we can detect. You basically, it's like taking an X-ray image. And what these do is they allow you to track the antibodies, right, and determine if they've attached to your target antigen. And they are very sensitive. So this is an example that I took from my research back in the day. And what I did here is I took a bunch of mouse poop and ran it on this gel. And then I took the blood serum from a mouse and I put it on top of the gel essentially. And I was able to detect antibodies in the blood to contents of the mouse's fecal matter, right? Which we haven't really got into it here and we probably won't much, but that's not a good thing, right? You don't want antibodies in your blood reacting to things in your gut, right? That could indicate an overactive immune system. Your gut is supposed to be isolated. So for any confusion that you may be experiencing, which you may be experiencing some, this is kind of the basic premise of all these immunoassays. And so you can do a couple of things. You can do direct test, you can do indirect test, but notice either way the labeled antibodies. So here, right, in this case, you have an unknown antigen with a known antibody, okay? So your unknown antigen is attached to some type of surface. Again, this can be a variety of things. I'm not necessarily interested in you feeling like you have to know exactly what they're attached to. I just want you to understand the basic premise of what's going on. So you take your antigen, you have a sample that has an antigen in it, you're not sure what's there, you attach it to a surface. You then take your antibodies that you know of, right? So basically you're taking a guess. You're saying, is the antigen that I'm interested in this sample? And I'm going to take an antibody where I know what it binds to figure out if the answer is yes or no. And so you take your antibodies that have some type of detectable marker on them. Again, that can fluoresce. It can cause a chemical reaction where it changes the color of the solution that you're testing this in, right? A lot of different possibilities. So in this case, you mix your antibodies with your unknown agent that's bound to this solid surface. And then you have two options, right? So after you've mixed these things together, you wash the sample. And if the antibodies have bound, even after the washing step, they stay attached. if the antibodies haven't bound after the washing step, right, they're removed. And then you're able to detect the presence of that antibody. And that would indicate that, hey, that unknown antigen, or, I have this big sample with a bunch of antigens in it. I can then say, yes, antigen X is in that unknown sample. Down here, there's no binding, there's no, we'll say, fluorescence. And so you say, okay, antigen X is not in that sample. Do you mean, Jennifer, do you mean like one class of antibody? Are you talking about like IgA, IgG, IgM? Or are you talking about like multiple antibodies for multiple unknown antigens? I'll check. Okay. So, I mean, you can really, so what you would do is, because you might have a lot of different, if you're curious about multiple unknown antigens that are in that sample, you would actually set up separate experiments for the different antigens that you were looking for in the same sample. If you're taking, and I don't know if this is exactly answer your question, if you were to take multiple antibodies to different antigens to put in the same sample, the system as designed like this would not be sensitive enough to determine which antibody is binding to which antigen. So you'd want to try to do one at a time. I'm not sure if that's what you're asking. If not, continue to harass me until I get right what you're trying to get at for me. All right. The indirect test that I was talking about. Oh, come on. Right here. This is, remember when I said an antibody to an antibody? So here, a similar concept, except now we know what the antigen is. So this would be like what I talked about in my experiment, right? My CBER protein. So I put that on a solid surface. I then take a patient's antibodies, right? Their serum, right? And I coat it on top of the antigen. I then do a wash, right? In this case, if they have antibodies in their serum to that antigen, it sticks. If they don't, it gets washed off. I then take an antibody that binds to the antibodies that are attached. And these now have the fluorescent signal or whatever signal you have on them, right? Nothing binds to this because there's nothing to bind to, right? And then I can get a reading again. Now, the way this works, in case you're wondering, well, what's this antibody to the antibody? So again, if we go back and we say, okay, my antibody that I'm using in this research experiment was generated in a mouse. All right, so let's say this antibody that you're using was generated in a mouse. So my secondary antibody is actually an antibody that is anti-mouse. And so that means that it will be able to detect or determine that this antibody is from a mouse, and so it wants to bind to it. Okay? Does that make sense? And so this is exactly what we just went over. This is a specific method called the ELISA method, right? Enzyme-linked immunosorbent assay. Ajax, do you guys do this in genetics? Do you do ELISAs in genetics?
Yes, they do like a mock HIV infection spread.
Okay. So if you did genetics, you should be kind of familiar with this. I assume that you maybe do this in some molecular lab. Do you do ELISAs there? Oh, okay. Well, so again, depending on what you do here, and if you do some research though with Dr. West or Dr. Cornett, you might do some ELISAs at some point, but this is the same principle, right? Look, here's the antigen, here's my antibody. So we have basically two different ways that we're going about it. You either know what your antigen is, you know what you know what your antigen is, or you know what your antibody is. And then this is just following the same basic principles of what we went over. Now, here's where Jennifer Buckner's question maybe gets answered kind of in a different way. So this is something called fluorescent activated cell sorter, FACS. Maybe you've heard of fluorescent cell cytometry, right? And what you can do here is you can take multiple antibodies to different antigens. In this case, we'd be trying to identify different cell types based on cell surface markers. And so if you remember CD4 and CD8 cells, right? If you wanted to separate those cells, you could take an anti-CD4 antibody and an anti-CD8 antibody and mix them together in the same sample. And what you would do is each one of those antibodies to CD4 or CD8 would have a different fluorescent marker on it. And you then pass them through this capillary tube that's getting hit with a laser. And you could detect based on the laser hitting the different fluorescent marker on the different antibody, and then you could separate them based on that marker. So in this case, the answer to Jennifer Buckner's question would be yes. In certain situations, you could take multiple antibodies that had their own individual markers, mix them into the same sample that would have different cell types, all right, and then sort it using a laser that could detect the fluorescence from that antibody cell interaction. Okay? It's a really, really powerful method that can be used to study different cell populations within a sample. Okay. Let's see. What do we got? What do we got? No. You mix the antibodies together and then you sort them. Right? So like if I took Alyssa's blood and I wanted to sort her cells, I would take her blood, I'd mix it with all the different antibodies, and then I'd hit it with the laser to sort them. Does that make sense, hopefully? Let's see. Keep going. So yeah, don't worry about that last slide. This is old school. If you guys have taken A&P2, you did this. This is still a really good, or kind of still a decently good way to identify samples. And so here you see the top one. This is agglutination, right? So agglutination is when you have antibodies, right? If you remember from what antibodies do, the idea of cross-linking, right? Cross-linking is agglutination. So here we can use this to identify different blood types. And so, Alanis, what type of blood does this person have? What type are they?
Which one?
This one right here. This person right here. This is blood sample from the same person. What's their blood type?
Anti-A-O-O. Is that right?
Well, okay. So
no.
no, I understand what you're saying, right? So what this is saying is the way this works is this is like a little card, and so on this card you have anti-A antibody and anti-B antibody. So if it causes agglutination or cross-linking, that means that antigen A is on the surface of that red blood cell. So this would be someone with type A blood.
Okay.
Does that make sense?
Yes.
Yeah. I'm sorry, that's not fair. If you didn't realize that the antibody is on the card, that's what it's referring to. All right.
That makes sense now.
This is also right a test. We'd have to talk to Professor Perry to see if this one's actually still used in research labs. But right, what you can see here is you can take antibodies to, in this case, they're looking at group A strep, right? And so you basically have manufactured these little beads that have group A strep antibodies on them. And so it's the same principle. You kind of force this cross-linking. And so if cross-linking occurs, then you know you have a group A strep bacteria. All right. And so, you know, again, these are kind of simple tests that are still used some, but if any of you have had ANP2, you also know that these might not be the most accurate things ever. And so these are not quite as common as they used to be. Right?
So for that specific example, if they were both like cross-linked and grouped together, would it be O?
No. What would it be?
I mean, it would be AB.
There we go.
Okay, sorry. Thank you.
So if there was no cross-linking at all, right, then what would it be?
Then it would be O.
There we go. So I know, Alanis, you just really wanted that type O blood bad. So there you go. You got it, right? All right. So let's go. Yeah, let's get into antibiotics. I also think this is a really interesting topic, just like vaccines, because antibiotic resistance is becoming a huge problem. And there's a lot of interesting reasons why it has become a problem. And there are a lot of people that are proposing some interesting solutions to the problem. All right. So one of the things I used to have you guys do is small reports on kind of figures in microbiology history, which I don't think is a terrible idea. I felt like a lot of students felt like it was tedious, but if you would have done, or if I would have had you do those reports, two of the big kind of, well, there's three of them, but we kind of make it into two categories. Folks that are really important in the antibiotic movement are Alexander Fleming, all right, who discovers penicillin in 1928. And then Chain and Flory, I'm sorry for forgetting their first names, are finally able to figure out how to produce penicillin on a large scale in 1941. All right, so you can see there's a good amount of time between the discovery of penicillin and its actual implementation as a medication that we are familiar with. Okay. This is also, these were the good old days where a lot of scientific discoveries were made on accident. So Alexander Fleming, I believe, went on holiday, right, vacation, and he had left some of his plates out that were growing bacteria, and Staph aureus specifically, and he noticed when he got back that there was a fungus growing that was inhibiting the growth of bacteria, right? And so that's how we actually discovered penicillin, right? We all probably know or realize that microorganisms are continually evolving, right? Again, we're not having the evolution debate, but regardless of where you fall, right? Microevolution, right, on a small scale is definitely something that we see and measure and track. Now, because microorganisms continually evolve, right, we have to continually modify our existing antibiotics, all right? So again, I mentioned antibiotic resistance. So we have to figure out a way to deal with those organisms. It's in the 1960s that doctors and scientists really began focusing on, okay, we have a good number of antibiotics that we know of already. Let's start modifying their structures to alter their properties against bacteria that maybe are becoming resistant, or see if we can modify their structures and have them interact with bacteria that they never could have before. All right. And so an example of this would be, again, penicillin, kind of the original antibiotic, which primarily goes after gram-positive. Again, we'll talk about why that is in just a second. Hopefully you guys remember. Oh, no, we haven't gone over that yet. We can talk about why that is in just a second. Sorry, forgot I'm doing things in reverse this semester. But it's been modified, right? And some of you maybe have taken ampicillin, and interestingly, it's from the same original structure, but the modifications to it now make ampicillin a good antibiotic against gram-positive and gram-negative bacteria. All right, and again, I know we haven't really talked about it yet, but I will give you a brief introduction as to what gram-positive and gram-negative bacteria are. So here you can see the penicillin family tree, right? So we kind of start in the 50s, and you can see here as we go up to the 1980s, we have a lot of different branches coming off of this tree. And so these are modified versions of penicillin. And some of you have maybe taken ampicillin or methicillin or amoxicillin, right, are relatively common that you may have had somewhat recently if you've been sick. So what do we want with our antibiotics? Micaiah, what should a good antibiotic do?
It should get out the infection.
Well, I'm sorry, can you say that one more time?
I said it should get out the infection.
Okay, good, right? So it should take care of the infection, right? That's number one, right? We want an antibiotic that kills bacteria. Perfect. What's the second thing that we want from a good antibiotic? And maybe think about if you have any family members, or if you have, and I don't know about it, that have undergone chemotherapy. That's kind of your clue.
Yep, she's right.
So can you say that again?
I was just saying that the person in the chat was right about how you don't want it to harm our normal cells, our normal flora.
Yeah. Who was that? Let's see. Jennifer Buckner. Okay, cool. Right, perfect. Right. So between Micaiah and Jennifer, that's it, right? A good antibiotic targets the pathogen. Boom, good. And hopefully doesn't harm the host. All right. And so, that's not really a realistic scenario in the sense of there's going to be some harm to the host. So ultimately, right, this point right here is you're looking for a greater harm to the microbe than to the host, right? So the basic premise is kill the bacteria before you kill the patient. Okay. So when it comes to kind of classifying a drug's ability to do this, this is what we identify as a therapeutic index, all right? And so when we're looking for a good therapeutic index, what we're trying to find is what is the lowest toxic dose, all right? So we want the toxicity to be as low as we can to still have the positive effect of taking out the pathogen, all right? And so that's typically what you are prescribed, right, as a patient, right? Through, research trials, right, they've determined, hey, I can give, right now I can see Micaiah's cat, right? I can give Micaiah's cat this antibiotic, right? And it's going to do the least amount of harm to her while still killing that pathogen, all right? So a high therapeutic index, that's what I was just saying, right? High therapeutic index indicates a low toxicity, so that's good. You want drugs with high therapeutic index. That means they're good at causing more harm to a microbe than to a host. Low therapeutic index medications do exist. They are very powerful, but they cause a lot of damage to the host. Typically, right, these are for topical application. All right, so I hope none of you have been tempted if you've ever had a topical antibiotic. When it says do not ingest, that's not just because it tastes bad, right? That's because that antibiotic is very, very toxic. Now, there can be some that are given intravenously, right, through an IV. But again, this would be done in a hospital setting where you could be monitored, all right? For the most part, and again, there's always exceptions to the rule, but I feel confident that a doctor is not going to send you home with a pill that's a low therapeutic index, right? Because that's risky, all right? But they are useful. All right.
Can I ask a question really quick?
You may, Alanis.
For things like radiation and chemo, would that be a low therapeutic index? Because it's hurting you more than, I mean, I guess it is helping you, but it's hurting you a lot more than like an antibiotic. I guess, is this just for antibiotics?
No, I mean, hold on one second. Sheila, did you get locked out? Hold on, guys. 1 second. I can't tell if my wife got locked out. Ugh. Ooh. There we go. You just got to witness me getting up on my own. So, Lannis, I would. Oh, no, it's unlocked. I don't know what they're doing. Okay. So, as far as, medications, I would say overall, that stuff, I mean, I would consider it low therapeutic index. Now, again, there's different variations, right, of chemotherapy. There's different things. Radiation has become more targeted. The basic premise of a chemotherapeutic agent is that it affects cells that are dividing more quickly, which is cancer. But yes, I would say they're low therapeutic compared to something like an antibiotic, right?
Yeah.
So that's a good question. So again, when we come to, how antibiotics are helping us deal with a pathogen, you basically have two categories, right? You have killing the pathogen directly versus inhibiting the growth. And both can be very, very useful, right? So another fancy way for saying inhibiting growth is to call something bacteriostatic. All right, and so it's static, right? It's not moving. Those of you that like physics stuff, all right? And so in this case, you're not killing the pathogen, you're just preventing it from growing. But that's still important because if it's not growing, it makes it much easier for the host immune system to clear that pathogen, right? So you're basically helping the immune system do its job, but ultimately it's the immune system that ends up clearing the pathogen, all right, if that makes sense. Your other option, right, killing, is something called bactericidal, all right? So in this case, you do kill the pathogen, and in this sense, the immune system is still working, right? It's still trying to do its job, but without the help of this medication, the immune system wouldn't be able to fully clear the pathogen. All right, so that's what I'm saying, right? You're doing what the immune system cannot do on its own. Again, it's still working, right? It's still killing some of the pathogens too, but if the drug wasn't present, it would not be able to keep up with the rapidly dividing bacteria. And one of the things that we'll get to here, I'll have to check, maybe next unit, is some of the things that allow bacteria to grow so quickly, right? Bacteria are growing at a much quicker rate. I'm sorry if I haven't said that. I'm sure some of you knew that. At a much quicker rate than your cells are. And so, these medications are trying to take advantage of some of that. All right. We also have ways that we can classify antibiotics, and some of you have probably heard these as either broad spectrum or narrow spectrum. All right. So broad spectrum is going to target a wide range of bacteria. You've heard me mention gram-positive, gram-negative bacteria. That basically has to do with cell wall and cell membrane structure, right? Some antibiotics, right, if they're narrow spectrum, might only target the cell structure or cell membrane, or the cell wall structure or cell membrane structure of a gram-negative, while some might only target the cell membrane or cell wall structure of a gram-positive. We even have bacteria called mycobacterium that don't have a cell wall, all right? So a broad spectrum antibiotic could go after all of those, all right? And these are really important in treating acute life-threatening diseases. So I'll just pick on her because she's the name that I see in the corner of my screen and the reduced zoom window. if Alanis comes into the hospital and she's super, super sick and they do a quick little blood test and they think she has a bacterial infection, they're not going to say, hey, Alanis, can you wait 24 to 48 hours while we identify the specific bacteria that's making you sick? All right. It's not a good general practice in keeping your patients alive. And so what they would hopefully do is say, hey, Alanis, take this broad spectrum antibiotic right now. We're going to give this to you. Then we're still going to try to figure out exactly what's making you sick. But in the meantime, right, take this antibiotic. It'll take care of everything for you. All right. Now, I put this in broad spectrum, but this could even be the same for narrow spectrum, except the effect is greater in broad spectrum, is it disrupts your normal microbiota. All right. Again, remember, we've kind of talked about and briefly mentioned that bacteria do a lot of good things in your body, or can do a lot of good things in your body. And so if you take a broad spectrum antibiotic that is killing all sorts of bacteria, it will also kill some of the good bacteria. And again, same concept in narrow spectrum, because there's good gram negative, there's good gram positive, and you could remove them from the system. In case you're kind of wondering, what this is like, some of you have probably experienced this. You may be in the group of people that have some type of GI distress when you're on antibiotics, and that GI distress is being caused because often you're killing some of the good stuff along with the bad stuff. Okay. And again, I've already mentioned, right, but for narrow spectrum, because it has a limited range, you need to be knowing what pathogen you're dealing with, or at least what class of pathogen, right? And so, gosh, I guess because she's the last one that talked, I'll keep on picking on Alanis. She's having a rough go of it here. So if Alanis has a gram negative bacteria that's causing an infection, if I give her an antibiotic to gram positive, Alanis is still going to have a gram negative bacteria causing her infection, right? Now, one of the things, and I think a couple of you are interested in pharmacy. I think at least Abby is. I don't know if there's some more of you, out there. One of the jobs of a pharmacist is to check the medication combinations that doctors are sometimes prescribing to their patients. And, sometimes it's not even necessarily a bad intention by a physician, but there could be this idea of you thinking, well, why don't I hit the patient with a bactericidal and a bacteriostatic, bactericidal and a bacteriostatic medication, or, mix and match, and that's going to be just awesome for the patient, right? And in some cases it can be, but in some cases it can be antagonistic, right? And so the reason that it can be antagonistic is that I briefly mentioned a couple slides ago. Some antibiotics, right, are good at killing bacteria because they interact with bacteria that are dividing. Okay. So if the antibiotic that you're giving to a patient works because it kills or targets bacteria that are dividing, but then you also give that patient a medication that's bacteriostatic that stops cell division, well, then it's kind of a wash, right? And so now your medications are actually opposing each other, and you're not really doing that much to remove the bacteria. Okay. So again, you have to be careful when you're combining some of these medications. You can also have an additive effect. If we were in class, one of the things that I like to do is I like to say an example of additive would be if you have a scale of antibiotic killing ability. 1 is least effective, 5 is most effective. If you took 2 bacteria that had a 5 on the killing scale and you added them together, Jada, what would their total killing ability then be if bacteria or if antibiotic A had 5 and bacteria or antibiotic B had 5? what would their additive killing power be?
I'm sorry, can you repeat the question one more time?
I can. So if antibiotic A has a killing power of 5, and antibiotic B has a killing power of 5, and you combine them, you add them together, right? This is an additive effect. What would their combined killing power be?
10?
10, right? 5 plus 5, right? So you're just saying, hey, this one does this job really well. I add it with this one. It does its job well. And so you kind of get that additive effect. All right, perfect. Synergistic would be, let's say A has 5 and B has 5. You put them together, and then the killing power is somehow 35. All right. And so, in that case, right, the killing power that you get is greater than the individual sums of them alone, right? And so that's a synergistic effect. So that's kind of a, that is a good, a potentially good thing for the patient, all right, if you can find something like that. But again, it's not that easy at just adding things together because they can oppose one another. All right. So adverse reactions to antibiotics, and this will be where we end. We'll end on the slide, and then we'll pick up hopefully in person on Monday. But again, some patients can be allergic, all right? And these allergies can be or start as small as just having a rash, right? So maybe developing hives to potentially developing anaphylactic shock, right, which can be life threatening. Again, we mentioned this in vaccines, right, because some of them are stored or preserved with antibiotics now. Maybe some of you are allergic to antibiotics. Then it's not a big deal because, again, we have a bunch of different antibiotics as long as the doctor knows to not give you that antibiotic. All right. Toxic effects. Right. Again, they can cause harm to the host. Where we really worry about this, especially, is infections that require long-term antibiotic use. So Lyme disease is an example, leprosy, tuberculosis. And again, we can talk about why that is, but they can start to have those negative effects on the body, which can lead to organ failure. You can even have inhibited production of red blood cells and white blood cells, which is not a good thing. And then one of my favorite, again, is microbiota dysbiosis, right? So removing the normal flora. So we've mentioned already, I think in class, the idea of C. diff. We have, especially an older patient, they come in with a bacterial infection. We treat them with antibiotics to make them feel better. It wipes out all of their good bacteria. And then you have this pathogenic bacteria, Clostridium difficile, that moves in to this newly established real estate. And then the patient is very, very sick. And then unfortunately, right, one of my favorite things with C. diff is you then say, okay, I gave him antibiotics. So this bad bacteria moved in, so now I'm going to give them more antibiotics, right? And that usually does not work overly well, right? So that's where fecal microbial transplants come in. Okay, so that's going to be where we end. I saw a question from Lauren about your exams. I just finished grading my A&P1 exams, and I've started on your micro exams. And so my hope would be to have those done this weekend. I would have loved to have had them done today, but I was struggling earlier in the week. So I'm sorry about that. But my goal is to hopefully have them done for you guys by this weekend. All right. Okay. I hope you guys have a great rest of your day and a great weekend. And I will hopefully see you in person on Friday. Okay, and I think I have all mastering bio issues resolved. If I don't, let me know. Okay, but I think I took care of everybody. All right, it was real. Thank you so much for your patience. I appreciate your willingness to work with me. Hopefully I'll be back and better than ever Well, hopefully I'll be back on Monday. I don't know, better than ever. All right. Have a great weekend. Miss you guys. And we'll get together soonish. Bye.